Okay, welcome to Introductory Genetics. In this lecture, we are going to be looking at some mutations, and we're looking at kind of large-scale mutations. So these are mutations on mainly chromosomes. So first off here, let's look at chromosomal mutations versus gene mutations. Okay. So as you can see here, we have gene mutations sometimes occur, as many of you probably know. There are just simple changes in sequence here. So as shown in a situation like this here, just simple changes will change how this gene is read, and different proteins will be created from that. And then if you look at the entire chromosome here, which is what we're going to be talking about today, you can have mutations where entire chromosomes get mutated. So chromosomal mutations. So a good way to look at these things is from And chromosomal mutations are defined by our ability to see mutations from a microscope. So, as you may know, we are capable of creating karyotypes of genomes for different organisms. And so right here, all these cells are arrested in metaphase, which is the phase where these chromosomes are most visible to the eye. We then use a microscope, and we are able to stain these cells, and we use the microscope to determine mutations. So from looking at this karyotype here, we notice that they're missing something down here. So in this case, they only have one X chromosome, so they're female and they're missing another X chromosome. So as you can see, this is a mutation. Classify these mutations as numerical, which is a rearrangement of entire chromosomes or genomes, or structural, which is a rearrangement of genes in a chromosome. So a little example here, structural would be, let's say, genes from here, these are and in such a case, one thing might occur where genes through crossing over actually, so some genes here may end up on this chromosome here, just through crossing over and many events in the stages of meiosis. And numerical rearrangement would be something like losing an X chromosome here, or gaining chromosomes here, and so on. So now we're going to get a closer look at numerical variation, and we're going to look at here aneuploids and polyploids. So right here, it's a karyotype, and I want you to see if you can figure out if this is an aneuploid or a polyploid based on your knowledge so far. Now, I didn't tell you what an aneuploid or a polyploid is. So in this case here, this is actually an aneuploid, and what happened here is that this individual has gained a chromosome right here. So they have 3 copies of chromosome 21. So this is simply a change in the number of chromosomes in the karyotype. A simple change in the number of chromosomes in a karyotype is aneuploidy. Okay? Now, aneuploidy can be monosomy or trisomy. And in the case where a monosomy or a so let's say if a trisomy, in this case, this is a trisomy here, there's 3. In the case that a trisomy is created, a monosomy is also created in a different gamete, and that would happen to be important for a different individual, or possibly an individual that never ended up coming about. In most cases, trisomies are actually capable of surviving. So in nature, we can actually see live trisomies. So in this case, trisomy 21 is Down syndrome, and people can actually survive with this mutation here. but that's not normally the case in monosomy. And exceptions for this case are usually have to do with sex chromosomes. So in this case here, if we ended up having two Y's or two X's here, what usually happens in many organisms is they only take on one of the X chromosomes information. So let's say females are very well known to have what's called as bar bodies, which they render one of the X chromosomes unusable, and they only take the information from one X chromosome. In the case where two Y's would be created, then the X chromosome alone would be used. And in both of those cases, there is a good chance that the individual would be able to survive. Now, taking a look over here, can you notice a bit of a difference between these two karyotypes? I hope you can. So in this case, this is polyploidy. And you can see how these chromosomes are homologous, and there's two of them here. So this one's homologous with this one. But in this case, every single chromosome has 3 in each situation. So this entire genome has extra genomic information that it actually does not need. So polyploidy then is defined as a change in the number of chromosome sets. And this is very uncommon in animals. It's actually more common solely in plants. And as we'll see later, the implications of having extra genomic information in a cell actually requires a larger nucleus and therefore larger cells. And if an organism has larger cells, it is usually larger. And this is a good thing for many fruits and vegetables. Larger watermelons and stuff are always better for the human population than smaller watermelons. So this has many applications in plants. So moving on here to aneuploidy. So how does aneuploidy come about? Well, as you'll see here, this is a normal meiosis event, and 100% of the gametes get normal number of chromosomes. So of course, this meiosis event can either be in a female or a male. Of course, if it was a female, this would not be the case here. 4 identical. Usually 3 of them get eliminated and 1 egg stands to be used. But this is the case in males for sperm. So in this case, yes, everything is perfect. That's great. Let's look at some abnormal activities here. So it's normal here, and then all of a sudden we have a split. And both of them end up going to this side here, into one cell. So this cell has no genetic information. It then has offspring that also don't have genetic information, and its offspring also don't have genetic information. And in this case, the cell has extra genetic information, and it gives rise to more cells with still extra genetic information in this case. Because as you can see, they should be haploid, but they're actually not. So 100% of gametes get a normal number of chromosomes. Yes, of course, because we want 1 here. We want a haploid, and that's not the case in any of these. So as you see, the issue right here is right here. 2 of them went to one side and 1 got nothing, and that has produced this case of aneuploidy. Another abnormal situation is in meiosis 2. So as you can see, that was meiosis 1, the first split. And here we have normal activity. The split looks normal here. And then these guys here are perfectly normal. But then we have an issue here. All the genetic information goes to this cell here, and this one loses out. And obviously these two are abnormal. And so now just looking at these a little bit more closely, these cells with nothing in them can give rise to monosomy in most cases, and the ones here with extra genetic information can give rise to trisomy. So you can expect that individuals that get trisomy have a better chance of surviving because they do have sufficient genetic. An interesting case here is presented, and that's right here. In this case, the individual that may come about from this, so we can say this is either a sperm or an egg, it will fertilize with a haploid cell, and what actually happens here is this could lead to uniparental disomy, which is the case where, let's say we have information come from a different cell and fuse with this to create a zygote. What can occur is that one of the chromosomes gets bar-bodied and does not function. And in the case that that bar-bodied chromosome is a chromosome that is not within here and is the one that comes from a different source, then the bar body will be the chromosome that is actually needed for genetic diversity. And in this case, the cell will be using simply both of these, and these actually come from one parent. So that's a quite strange case, which leads to less genetic diversity. Now in the case where you do get extra information, so here, in this case here, and in this case here, where you end up with a trisomy, gene dosage is actually a big factor in survival. So in the case that you are capable of living with trisomy, proteins function as this genetic information can code for proteins that function as complexes. So this is just a well-known thing that most proteins come together from different polypeptides, and these polypeptides come about from different genes, and these genes can come from different chromosomes. So these proteins function as complexes, and the protein complex has these subunits encoded from different chromosomes. So quality control ensures that proteins function normally and abnormal ones are destroyed. In trisomy, too much supply of one complex occurs, and the body needs to make extra destructive mechanisms to destroy proteins. In monosomy, too little would be created, and proteins would still need to be broken down, or not enough would be created. So I hope this makes sense. In the case of trisomy, the protein complexes that are created come about from different organisms, chromosomes, and what can occur is that if a gene is needed from a chromosome that is actually trisomic, then there will be more genes for this coding for this complex. And so not all these genes can be used, and many of them need to be destroyed because there's just too many of them. And that can obviously wreak havoc on the cell, which can be a big issue. And so the only trisomies that can survive are trisomies of chromosomes that have very few genes. So a common case here, and especially this is true for humans and in many organisms, the lower number, the bigger the chromosome. So we know that we know that chromosomes are ordered 1 to 23, because that's how many we have. Then, in that case, the lower number, so let's say 1 is a very low number, the lowest number, 1, has the biggest chromosome, and that's how they're ordered. The highest number has the smallest chromosome. Now, this is not correlated to the amount of genes on the chromosome, but it is slightly correlated. So in historical cases, we have come to realize that many diseases can come about from trisomy because they are actually capable of surviving. So things such as trisomy 21, Down syndrome, trisomy 18 have been reported. These individuals, their gametes, and through mitosis, their cells were not aborted, and they've been able to survive because the chromosomes that they're trisomic for are not coding for a lot of genes, and therefore they have been able to survive. So trisomy is usually, well, most commonly caused from a mutation in meiosis 1 in the female. For females, eggs are produced prior to birth, and all eggs are suspended in meiosis 1, so in prophase. And when it sits, it can get mutated, and it can lead to disjunction. Also, degeneration of spindles and other apparatuses within the cell that help to undergo these division events in meiosis, they can also begin to degrade after so much time. So you can see how a female that reproduces at 40 years old has a higher chance of having offspring with a trisomy or a monosomy, any type of aneuploidy, than does a female that's reproducing at 20 years old. And there is statistics to show that this is actually true. So men can also get errors due to replication. Men make trillions and trillions of sperm throughout their life, and there's always some number of improper replications that men go through. And in the chance that one of these improper replications ends up coming together with the egg of the female, then there can also be issues there. So now moving on to polyploidy. So as shown before in aneuploidy, in the case that one of the parent gametes ends up as 2N, then when they form with another gamete, then the individual can become 3N, or 4N if there is another mutation that occurs. And in the case that like in this case here, both parents are 2N, and they've created a 4N offspring. There could also be the case where this is 2N and this is haploid, and they could create a 3N offspring. So that creates polyploidy. So this is much easier for plants to do because they reproduce with pollen and not not how humans reproduce. There are two types of polyploidy. So there's autoploidy and alleloploidy. And autoploidy is chromosome sets from a single species, and alleloploidy is chromosome sets from 2 or more species. And autoploidy can result in genetic mosaicism, and the mosaicism I'm not going to go into detail about today, but the degree of mosaicism can be determined by how early mutations occur in mitosis. Now lastly, just looking here at chromosome structure variation. So as you can see, there are chromosomes here, and what chromosomes are capable of doing is cross over genetic information. So as shown here, the B on this case is moving over onto this side. It comes over here, and the V ends up here, and the W ends up on this side, and ends up here, and that's the case. They have swapped information. So these duplication and deletion events can be unbalanced or balanced. Unbalanced is a gain or loss of genetic material, and balanced is no loss or gain in genetic material. So obviously balanced, in the situation where it's balanced, the offspring have a very good chance of surviving. They, in most cases, do survive. And in unbalanced, there is the possibility that the individual may not survive. It really depends on the severity of the duplication or the deletion.